Okay, it is 9:15, so welcome everyone. Thank you for joining us at the MASERC Research and Management Showcase. Let's see, right now you are in Web Room 5, so we're about to welcome Josh and Chris to talk about their eDNA project. I will have them introduce themselves in just a minute. I'm going to give a couple of quick housekeeping items. If you have questions during the talk, you can put those into the Q&A, and please try to use that Q&A button instead of the chat. We'll be able to help clear out questions that way. I believe there's even a way to upvote there. So that'll be your best spot to put your questions for Chris and Josh. If you actually put them in chat, it's no problem. I'll see them. I'll try and push them over for you. There is currently an auto transcription going on. If you'd prefer to not see that, you should be able to hover over that as well, and you can shut those off. All of these presentations will be recorded. They'll be uploaded to YouTube after the showcase. Give a little bit of time to clean up all the talks and everything and get them ready and loaded. But you'll get a notification once those are ready. And with that, I think we're ready to welcome our next presenters. Chris or Josh, I forget who's presenting first. You should be able to take over screen sharing and put your presentation up, and I will turn it over to you. Thank you.
Thank you. All right. Okay, I should have gotten the cover slide up. My name is Josh Dumke. I have been working with the Natural Resources Research Institute for about 10 years doing aquatic resources science on invasive species and fisheries. And this project, we've been working with folks at the University of Minnesota main campus down in the Twin Cities. So I'm the project PI on this project, optimizing eDNA monitoring for multiple AIS. And Gretchen Hansen is leading a team down in Twin Cities with a grad student, Chris Rounds, who is going to be presenting with me today. And Dr. Chanlan Chun up here at NRI is leading the microbiology work, doing a lot of the DNA processing in the lab, and is advising a grad student up here, Anna Toch, who has a poster on this work that'll be in the poster session. And we're all being helped by Dr. Eric Larson at University of Illinois, who has a lot of experience doing eDNA work and gave us a lot of tips and tricks on how to avoid cross-contamination in the field and in the lab, and types of things that we should focus on for making sure that our samples are have good quality control and everything like that. So I'm going to get started on talking first, and then Chris is going to talk after I am done, and we'll flip back, and then we'll have questions at the end. So invasive species in Minnesota, we have a map here showing where we know invasive species to be present in Minnesota right now from the DNR Manifested Waters list. And the basic premise is that we need to know where invasive species are in order to slow their spread. One of the problems is that early detection using traditional methods requires substantial investment, and that means time, but also money, or trained personnel, where the amount of effort that can be put forth is limited by how many dollars and time you have to put towards looking. And that limits the number of lakes that can be surveyed. And typically it limits the lakes that are being surveyed to ones that are really popular, that are in public eye and people notice and recreate at frequently. And a lot of the lakes that kind of get dropped off the list for being monitored might be more remote, they might be visited less, but they may still have just as high of a chance of having an invasive species present that we don't know about. And even if you're doing these surveys, the physical detections may still lag years behind invasive species establishment, because doing traditional surveys, you actually have to capture an organism or a plant. They might be established at very low abundances for quite some time before they're actually detected. And this means there's missed opportunities for preventing the spread of invasive species. So we think that environmental DNA can help, or eDNA for short, because eDNA can screen for many species from each sample. So our technician, Colin Krochok, is holding a sample right there. That bottle of water can have the DNA from all organisms that are living in that lake. And eDNA may perform better than traditional methods when AIS are at low densities. And that's mainly because you don't actually have to capture the organism itself. You're capturing mucus or skin cells or gametes or anything left behind by that organism, even exoskeleton, when organisms shed their exoskeletons and things like that. There's DNA in the water, and so you don't actually need to capture the organism. eDNA sampling is relatively inexpensive, and that it is also conducive to widespread monitoring, like use by lake associations or citizen scientists. So as this photo shows, this is a sample. So all that's really required is about a minute of training just to talk about how to handle the sample bottle and collect the sample and keep things from being contaminated. But a pair of gloves, a bottle, a label, and a baggie are basically the materials that are required. Put it in a cooler when you're done, and that's a sample. So how do we maximize AIS detections using eDNA? Well, to get the most out of eDNA monitoring for multiple invasive species, we need to know two things. When and where should we collect water samples? And that's the first objective of this project that Chris is going to talk about next. And what combination of methods maximize DNA yield? That's what I'll talk about after Chris is done speaking for a few slides. And we'll give it away to Chris now. Chris, I should have given you remote control of my screen. So see if you can flip to the next slide. There you go. And you can take it away, Chris.
Sorry, I was muted when you gave me remote control, so I could not talk there. So if you could give me control again, that would be awesome.
Okay.
Okay, so yeah, just talking about objective one here. So this is really focusing on kind of when and where we should be focusing our effort in collecting these water samples. So for this, we are collecting and analyzing samples from Minnesota lakes to detect presence of, we're focusing on 4 AIS. So that's zebra mussels, spiny water flea, rusty crayfish, and common carp. And so we're looking at how seasonal timing of sampling, the influence of different physical and chemical lake characteristics and locations of samples really influence our detection. So talking about seasonal timing of sampling, you can see on this graph here, we have common carp kind of have this real large peak. This is all theoretical data from one of our collaborators, Eric Larson, but has this large peak when they would be spawning in the spring. So kind of working in how these invasive species life histories would kind of change their probabilities for detection. So the and then, you know, they have kind of this maximized zone right here, noted in this square box, where all 3 of these species, once again, hypothetical and not the species we're using, but they kind of have like a maximized kind of ability of detection. So at this point, you would have to take the least amount of water samples to be able to have a 95% confidence of detecting these species. And so that's kind of like, you know, one thing that we're looking to get out of this. When can we like go to a lake and take a water sample and be able to have our best ability to detect all of the species that we're looking for in that lake? Let's see if I can move. All right. Ooh, wrong way. Okay, so on our kind of study design and sampling for objective 1, so we're visiting around 20 lakes 5 different times throughout the open water season. Each of these lakes has 1 to all 4 of our AIS. So Lake Fallon is a Ramsey County lake that Ramsey County has been very helpful in sampling. And this has 2 of our AIS. So this is rusty crayfish and common carp. And so from this lake, we would take 14 different surface water samples. 10 of these samples are used for eDNA. So at each of these kind of points, one of these red or black points, we would take a water sample for eDNA. And then at each of these black points, we would take a sample for analysis of major cations and anions. So that allows us to see what kind of chemical characteristics of the lake maybe are inhibiting our ability to detect eDNA. And then we also use field blanks, and that is really just to make sure that we don't have cross-contamination in our samples. So our field blanks are pretty much just DI water, deionized water that obviously should not have any kind of DNA in it at all. And then we do the same kind of DNA extraction process for that blank. And so once we go to a lake and take all of our samples, we bring them back to the lab for filtration, preservation, and then later on DNA extraction and quantification using qPCR. Current progress that we're at for Objective 1 right now, we have finished 4 sampling events for 13 different lakes this season. So we still have 1 more sampling event in 2021, and this will occur after fall turnover. So that's kind of when temperatures cool down, lakes will mix again. And that's kind of thought to maybe have influence our ability to detect invasive species, because all of this water is completely mixing throughout the lake. DNA is just being spread throughout this totality of the water column. And then, yeah, there's just, I guess, more even spread of DNA throughout the lake. So currently we're working on processing all of our eDNA and cation anion samples. That's over 750 in total, not including this last sampling event that we have. So a lot of work for us this winter off season. And in 2020, we will be going out to 8 more lakes to do continued sampling. And then what we hope to have for our application of findings. So our kind of number 1 goal of this, I would say, is to have a plain language guide, kind of a guide that we can distribute that is really like not in the weeds of eDNA, but kind of anyone that is looking to implement an eDNA monitoring program would be able to use to be able to detect AIS on their lakes. So this would be kind of for lake associations, maybe more county governmental or institutions. And so by doing, or the way that we're going to do that is kind of see what physical and chemical lake characteristics where eDNA works best. So, you know, there have been thought like that smaller lakes might be, the spread of eDNA throughout the lake might be greater. So certain lakes might be more conducive to kind of this, this type of sampling. And then there are certain inhibitors. I just got a question that I should mention. Good point. Cations, anions are like chemicals pretty much that will like break up. So like salt, NaCl, sodium chloride will break up, and the Na will be a cation, a positively charged chemical, and the chloride will be a negatively charged anion. So different things like that. And I've seen things that maybe, you know, certain anions can inhibit the ability to detect invasive species. So we're kind of like looking to see what lake characteristics really eDNA can work best in. And we're going to be using a detection probability modeling framework. Some people from the USGS created this amazing statistics package called eDNA occupancy that can really use a variety of covariates like that we are collecting to be able to piece apart kind of when or how these eDNA detections are working and when they aren't working. And with that, that's the end of my slides, and I'll pass it back over to Josh. Look forward to answering some questions.
Thanks, Chris. So for objective 2, we're exploring a little bit more about how to take samples and how to process downstream lab work that are going to be optimizing your ability to capture DNA from water and quantify it. So it's broken into 2 questions. Field methods are going to be expanded on in the next few slides briefly for each of these topics, but basically, how much water should you collect? Doesn't matter where you collect that water, from near shore to offshore, but also offshore, surface, and deep. And how does AIS presence correlate to eDNA detection? So in the places that we collect eDNA water samples, we're actually doing some traditional invasive species surveys and looking to see if our target organisms were in the location we collected water. And lab methods, once you collect that water, it doesn't matter what pore size filter you use to filter it and capture the DNA. And does preservation and extraction method matter? There are some easy things you can do and some more involved lab methods you can use that might work better, but is that extra effort going to pay off? And does quantification method matter? Most of our samples are being processed and quantified with real-time qPCR, but we're sending a subset of our samples for a droplet digital PCR, which is a more sensitive method. So to answer some of these questions, we selected 5 lakes with our target invasive species: Lake of the Woods, Vermilion, Shagwell Pike, and St. Louis River. These are all in North and Northeast Minnesota. They all have different abundances of the invasive species that we're targeting in this project. From each lake, we collected a lot of samples, 125 samples collected per lake. Most of them are for eDNA. We did take some samples for cations and anions and for field blanks. And in the map of Pike Lake on the bottom, you can see green and red dots. And these sample locations are split up by whether they're near shore or offshore locations. And we do a little bit different things, whether it's a near shore or an offshore place. And the sample locations that we took eDNA samples, we had multiple paired eDNA test groups. So we're collecting a lot of bottles of water from the same place at the same time, and then all those bottles of water are treated into different test groups. So we have paired test groups where most of our sample sizes are 75 samples per test group. And we're looking at the same species as objective 1: zebra mussels, spiny water flea, rusty crayfish. Common carp, we are only processing for the St. Louis River estuary. That's the only objective 2 lake where they are known to occupy. And eDNA collection points are paired with traditional invasive species surveys. And we do a little bit different traditional surveys, whether they're near shore or offshore. So for the first question, how much water to collect? Why are we even asking this question? Isn't it always better to get more water? Getting a 1 liter water grab is 4 times as much water as a 250 milliliter grab, but it also takes up more space in your cooler. It weighs more. The bottles cost about 3 times as much as a 250 ml bottle. So we wanted to test whether or not taking a 1 liter grab is worth the extra effort to get these samples and move them around, or would it just be better off to take more 250 milliliter grab samples? You can get more of them into a cooler, and it's a lot easier to filter, to be honest. Some of the 1 liter samples, filtering them, the filters would clog before 1 liter water volume was passed. So we'd have to set up another filter cup and split that sample among several filters. So we're asking that question with the application of making recommendations that are that make sense, they work, and would be easier for field collection and filtering downstream. It doesn't matter where we collect that water. I mentioned we're taking near shore and offshore samples. At the offshore, we're also taking deep samples. So we're getting a water sample at the surface, but we're also sending down a Van Dorn, which has these doors on it that when you lower it near the lake bottom, you send down a weight down the rope that hits a trigger, the doors close, and it encapsulates water from near the lake bottom. Then we bring that up and take that water as a sample. And that could be important because some of the invasive species are inhabiting lake bottoms, like rusty crayfish. They might not be nearly as active near the lake surface, so perhaps their DNA is more concentrated near the location that they're living in. So we wanted to test whether or not it's worth the extra effort to have a Van Doorn and collect samples from near the lake bottom, because it is about a $600 or $700 device. It's more futzing around to decontaminate it and clean it between sample collection points. And it's just more to deal with. So do you have to go through that extra work, or are surface samples going to be completely adequate? So that was one of our questions. And how does invasive species presence correlate to eDNA detection? We're asking this question because in a lot of our lit review papers would have some unknown reasons why their eDNA samples came back with non-detections for target species that they thought were present or should be present. And we correlated, we decided to correlate whether or not these invasive species were present at our sample locations to help explain our eDNA results. So if we get any results that we think don't make sense, at least we've got some traditional surveys to look back and say, well, yeah, we did put some effort into detecting this species at this spot and we didn't find them. So maybe the non-detection really is because they weren't in this location and therefore their DNA wasn't there, or it was in such low abundance it couldn't be detected. So it's basically to correlate with our eDNA presence and absence results and explain some of our presence and absence from the eDNA samples. So near shore places that we took eDNA grabs, we were doing some snorkeling or shoreline searches where you go around with a net and turn over rocks and logs and look for adult zebra mussels, and several times capturing rusty crayfish as they scurried away. We also set overnight crayfish traps to capture rusty crayfish, and did horizontal plankton tows to get zebra mussel veligers or spiny water flea that might have been near shore. And at offshore locations, we did vertical zooplankton tows, as well as horizontal zooplankton tows. And we would send down a remote underwater vehicle that we borrowed from Minnesota Sea Grant to look at the lake bottom, again, to see adult zebra mussels or rusty crayfish, and any other incidental invasive species like Chinese mystery snail. We would see those pretty frequently too. If water clarity wasn't good enough for that, we would send down a Petit Ponar dredge and pull up samples of the substrate to see if it was really mucky, silty, soft stuff that zebra mussels would not be attached in. And if it was firmer and native unionid mussels were living in that sediment, often we would find zebra mussels attached to those native mussels on the shells that are exposed. So that was how we were looking to verify if zebra mussel adults were present near the bottom when we were taking our offshore samples. And in the lab, does filter pore size matter? We looked at 1 micron and 5 micron filters. The 5 micron has larger spaces between the mesh, so material and water flows through it a little easier. And you can see on the bottom figure bottom image what a filtered water sample looks like. And there's DNA on there, but there's also algae and suspended sediments and anything that's in the water column. And so 1 micron filters do tend to clog up faster than 5 micron, but we wanted to see whether or not there was going to be a benefit in choosing one filter pore size over the other. And all the filters we used are cellulose nitrate material. Does preservation and extraction method matter? All of our samples, the majority of our samples are preserved in 95% alcohol after they're filtered. We take that filter, fold it up, and put it in a microcentrifuge tube. And then the extraction occurs with a HiGen DNA Easy Power Soil Pro kit. And that's a pretty easy thing to do. All of these things, the centrifuge tubes, the HiGen kits, and alcohol are pretty easy to get and order off the internet. And there's another method of preserving in CTAB and doing a chloroform extraction, which requires extra lab steps, a little bit more know-how. It's not quite like a packaged kit that you can order like the HiGen DNA Easy kits, but it might perform better. And so we wanted to test whether or not doing the extra lab effort was going to produce results that were measurable. And we also added in a silica bead desiccant preservation test. And this one was a little bit different. There's a syringe that you filter water in the field with. So the white cap is a housing that includes the filter. You fill the syringe with lake water, push that water through that filter housing. The filter captures the material and you put that into a baggie with silica desiccant beads. And the drawing out that filter can preserve the DNA. And we wanted to test whether or not that was a viable method and how it compared to our other preservation methods. Because if that does work, then samples could be sent through the mail. And so that was one of the reasons why we included this kit. It also makes it easier to do longer excursions where you don't have a cooler or you can't carry a whole bunch of water, like if you were traveling through the boundary waters. It might make AIS monitoring more accessible that way. But we wanted to test how effective it was first. And does gene quantification method matter? For the most part, we're using real-time PCR, which takes your sample as one measurement per invasive species that you're looking for. And we're going to compare about 125 samples with ddPCR, which takes that same volume of sample and splits it into droplets and basically looks for presence and absence thousands of times for a sample. And a computer does some statistics about how much DNA likely was in that sample for the species you're targeting. But the point is, it's a more sensitive method. You have much lower detection limits of detection. So in theory, during early invasion of invasive species, where their DNA may be low, ddPCR may pay off. But we want to test that in comparison and see if the extra expense and the difficulty in getting this method conducted, because ddPCR is a separate machine and not every place can do it, whether going through the extra effort is going to pay off. For objective 2, our current progress is that we've gone through and done all of our sampling for this year, and objective 2 fieldwork is complete. We've collected 625 samples, most of them are for eDNA. And our traditional AIS searches yielded many positive detections at eDNA sample points. So from crayfish trapping, we got rusty crayfish. We also saw rusty crayfish with the underwater drone. Horizontal plankton nets, we got spiny water flea. From Ponar dredges, we pulled up mussels that had zebra mussels attached, and shoreline searches and snorkeling, there were several pieces of wood, rock, etc. with zebra mussels attached to them. So we were able to confirm that a lot of our eDNA collection points had some of our target invasive species at them. So I'm interested to see how those results correlate. So in summary, most of the 2021 fieldwork and sample filtering is done. Samples are preserved and waiting in NRI for extraction and quantification of target AIS genes. And by project end, we hope to know which lake types are more suited for eDNA as a AIS detection method, what time of year to collect water samples for eDNA, where to collect those water samples in a lake, and which of our tested field methods work best for capturing eDNA in the field, and which of our tested lab methods are most sensitive at recovering and quantifying DNA. And the point there is that if the DNA is not very abundant in the environment, you want to choose the methods that are maximizing your ability to capture and quantify that DNA. Our deliverables for this are to create method recommendations for the field application of eDNA when monitoring invasive species in Minnesota. So managers, specialists, and non-government organizations have guidance and confidence when using eDNA as a monitoring strategy. And to that end, we would create an eDNA best practices and where to start guide, which will describe our findings and recommendations in plain language. So we want to learn what are going to optimize what methods would optimize eDNA as a tool to look for invasive species as a monitoring program, and then disseminate those results and findings to people that can actually use them in application. So with that, I want to thank everybody for joining us. My email and Chris's email are up. So if questions don't get answered, send me an email. I will also forward any emails that are specific to lab methods to our project team, Chanlan Chun or Eric Larson, because I do not work in the genetics lab. So if there are very specific questions about that kind of stuff, I'll have to pass those on, but I'll do my best to answer everything else. And thanks to our funders, affiliations, partners, and supporters. As Chris said, Ramsey County has been helping with some field sampling, and Sea Grant gave me a drone to use for the summer. So we appreciate all of the help that we've been getting. And thank you.
Josh, I don't know if you see in the Q&A, there's a question that you might be able to answer about starry stonewort.
I see that. No, we are not looking at starry stonewort in this project. We could expand in the samples that we've collected this year. We've collected them in a way that would allow us to use to do metabarcoding later. So we could add in searches for other species from all of the samples that we've been collecting for this project. We're looking for 4 species, but the DNA from several species in theory could be on those filters that we collected. There's another question about technology eDNA moving fast. Yes, and regard to cost of sampling, the speed of getting results, how do you see us changing in the next 3 to 5 years? I don't know exactly how cost is going to change in the next 3 years. It is more cost effective to run more samples than a small number of samples. Kind of the cost per sample decreases as you get more samples, because some of these machine runs can handle a 96 well plate at once. So there's going to be cost reductions, I'm sure, coming in the future with some of the equipment like ddPCR, if it starts becoming more and more common that universities or private companies have the ability to do ddPCR, that could become more cost effective. And changing the next 3 to 5 years, honestly, I don't know how the science of eDNA will change in the next 3 to 5 years. I am probably not the best person to answer that part. Our project partners, Eric Larson or Chanlan Chun, might have a better idea about that. But I see eDNA as a tool for invasive species monitoring probably in the next near future, being used as an important screening tool. So you go and get a bunch of samples. If you get positive detections for the eDNA, then you know where to send a team of people that are trained in invasive species traditional surveys to go and look for it and do that verification. I see that as probably the next 3 to 5 years is how eDNA will work as an AIS monitoring tool. Timeline for making guidance available. Our project ends next December. Report requirements are a couple months after project ends. So I'm hoping that by what would be 15 months or so, because we have to get through all of our sample, our needed sampling next year for objective 1. So we'll have preliminary information probably in about 12 months from now, but it probably wouldn't be finalized until project end. And we'll probably have a period where we're working very closely with MASERC staff to make sure that the guide is formatted and correctly and placed where people have access to it. So that's what we'll end up working with. I'm guessing that it'll be after our project ends, after next December. But we'll have some preliminary guidance available probably in the next year. And we'll probably share that at the next showcase. Okay, another question. There are many methods using eDNA. Yes, yeah. When do you think we'll have a standardization of an eDNA method? Honestly, standardizing, I don't know. That is very true. There are many methods. You can choose probably a dozen different filter materials. We didn't even investigate filter material. There are, probably a dozen pore sizes that people use. We chose the most 2 common reported in literature. when folks started with a 1 micron filter and had them clogged too fast, often they say they had to switch to a 5 micron halfway through their project. And so that's why we chose 1 and 5 pore size filters. So we did not test everything. We tested kind of the most common practical things that we saw other people reporting that they were using. And we wanted to see how it was going to work in Minnesota, especially when our late July sampling event, several of the lakes that we visited had enormous algae blooms. And how does that affect your water volume you choose and the filter you choose? Because that algae clogs everything up. And so we wanted to test some of these things that might have been used throughout the United States in a Minnesota setting and see how things worked out. So I don't know if we will ever have a standard eDNA method that works everywhere. In several years and several more projects, I think things will get narrowed down. And it would be great to have a standardized method. I don't know when that will happen though. Okay. Do you have preference habitat identification for zebra mussel based on eDNA? That wasn't a part of this project. We selected lakes that we knew had zebra mussel or were not known to have zebra mussel. And so we don't exactly have any part of this project that was, finding what habitats zebra mussels are selecting. I'm guessing that's for either lakes or for substrate areas within lakes based on substrate. So we didn't work with that. No, that wasn't a part of this project. We're basically testing the method of what makes eDNA work as a tool based on time of year and how you take samples. And another question is evaluating the ABIS filtering technology being incorporated into this project. The ABIS lab had created a sorbent that we were talking about using when we were devising this project, but it seems like due to COVID-19, some of the production and manufacturing got delayed. And so it wouldn't have been ready in time for us to use. So it isn't being used in this project. Oh, okay, here's a question. Has previous research or work solidified the primer needed for each species? Any concern with amplification? As far as I know, we are using the primers that are reported in other literature. We have a probe for rusty crayfish that Dr. Eric Larson recently worked on that should help be more specific to rusty crayfish. I don't know. I cannot answer that question effectively, whether or not we have concerns with amplification. So I'm not the best person to answer that one. As far as I know, in discussions with our project team, with the folks that actually know about this stuff, Chan Lan and Eric Larson, it seemed like they weren't concerned, and we'll see as we start getting through our extractions and qPCR, whether or not there's anything that we need to discuss, any concerns that we have. Right now, all of our samples are preserved. Extractions have started on maybe 10% of the samples that we have, but we're just ending our field season and we're getting into the lab season now. So hopefully we don't end up with concerns, and we'll find ways to work with that challenge if it does happen. Okay, another question. If by chance not detect a targeted species, how can we be sure that it doesn't come from the method which is not adapted or inhibitors? So that's part of one of the reasons why for Objective 2, we're doing traditional surveys also to help explain whether or not we have any issues with our eDNA as a detection method. We're collecting the cations and anions from the lakes as we take eDNA samples. So hopefully we have some idea on whether or not we have inhibition due to lake chemistry. Yeah, that's another question that our Pima folks, Chan Lan and Eric Larson, would probably be more able to answer than I am.
I would
So-
say as well, like, so based on previous literature that has kind of used eDNA to detect any of these species, they've kind of used like really similar lab and field methods, maybe besides preservation. But you know, other people have had good results with this. And when we're talking about inhibitors, I wouldn't say it's going to be like something where all across the lake we will get absolutely no detections for something like you know, zebra mussels. It's like you know, water chemistry varies within a lake, so it might be that if you have a certain amount of ammonia at one spot or something, that's completely made up, but that could inhibit it. But we still expect to see detections. You know, like we don't expect it to be inhibited all throughout the lake, but that's something that we will see from after we get some results back.
Right, and that's, Chris brings up a good point. That's important to note that our sample unit is a lake. So we might get non-detections at individual points, but if we get positive detections at other points, then we're still getting a detection for that lake. There's another question, which is the most potent inhibitor in the water? I don't know. I'll be honest with you. I don't know the answer to that.
Yeah, I don't know the answer to that either. I recently read something that said chloride might be, but that would kind of make it so that in saltwater it wouldn't work at all. So I don't know if that is actually a good inhibitor. I don't think it didn't really make sense to me, but that's something that we will have results on potentially next year. So for objective 1, we are doing presence absence and our ability to detect our 4AAS, but objective 2, Josh might be able to touch on more.
Yeah, on the objective 2, we are doing rough, not necessarily like measuring population density, but we are getting kind of rough classifications on whether there were few, many, or, you know, it completely carpeting the bottom. If, you know, we're looking at zebra mussels, there are several places where on Pike Lake, zebra mussels were carpeting the bottom of the lake, versus, you know, you search on the shoreline for 8 or 9 minutes and you find, you know, 3 of them stuck onto a piece of wood. And so we are making notes in our field data sheets about how common each species was. We will be able to actually do densities when we look at the zooplankton tow, because that's sampling a known volume of water. So we'll know how many zebra mussel veligers are in a unit of water, and same with spiny water fleas. So we'll actually be able to quantify the zooplankton data, but most of the other stuff is kind of classified as how abundant they were. We have, you know, numbers of rusty crayfish captured in each of the crayfish traps, but it's a little bit difficult to use that because the traps are baited and it can draw rusty crayfish in from a greater distance. So the actual point that you do your sampling, you don't know how many of those rusty crayfish came from that immediate location versus rusty crayfish traveling along the lake bottom, following the scent of the bait and coming into that area. So that's why we're doing snorkeling and trapping, because we can confirm that rusty crayfish were present right where we took our sample, when we took our samples, and then we do the trapping, and it can actually get abundances. We can get numbers of them. So we're kind of doing both for crayfish at least. Do you think we can determine the number of individuals of a given species by quantifying its eDNA? At least right now, I don't see that. There are periods in the summer where there will be super high pulses of DNA, like when zebra mussel veligers are released in the water column, or when carp are spawning, or maybe when rusty crayfish are molting. There are these spikes where we would expect a lot more DNA to be in the environment than just the organism living in that environment without having one of these biological events that creates a lot of DNA. It's possible that we could find strategies to sample at a time of year when we know that they're not biologically super active. Maybe then quantifying their genes would be more applicable to quantifying number of individuals. But at least right now, no, it doesn't seem like we can do a very good correlation of eDNA sampling and quantifying DNA and then turning that into how many individuals were there. So right now, it doesn't seem like that's possible to do.
Megan has emailed us and said that her controls have been locked. So I think we
sharing.
Yeah. I don't know if that helps.
Okay.
I think we can move to the next room. It is.
Yeah, I think that we are past 10 o'clock, so we will be done. Thank you, everybody.
Thank you, guys.
Thank you both for that wonderful talk. We already have some questions coming in, and I would just like to remind the other participants that if you'd like to ask a question, please use the Q&A feature at the bottom of your screen. If you don't see it, you may have to hover your mouse in order to find it. So our first question is from Nicole, and they are asking, does eDNA sampling test for aquatic plants? For example, Eurasian water milfoil or starry stone wort.
It's possible to. We didn't focus on that in this project. I think because not as many plants have good markers or primers available, and I don't know if that's being worked on by other groups. But from my understanding, one of the limitations was good genetic markers to use for many different plant species. Yes, and our samples were collected in a way that would allow metabarcoding or later investigation from our same samples that'll be archived. So we could always go back and search for things from our samples that we have now and look for other things.
Great, thank you. And Steve also had a very similar question asking about eDNA being used to identify non-native aquatic plants. So that has already been answered. While doing this project, have you identified any additional invasive species on these lakes? Or have you not run all the samples yet to know that?
We haven't run any samples yet to know that, and we haven't in our traditional AIS surveys, we haven't come across anything new as we were doing our searches. So not yet.
That's good. That's a good thing for now. We have another question asking, do you use a preservative in your water samples?
The water samples are raw water that is kept cold on ice in a cooler until being filtered.
Yeah, and we kind of follow like best practices is like keeping them out of the sunlight, you know, keeping them on ice, filtering them pretty much as soon as possible to preserve as much DNA as possible.
Our next question is from Mike, and they're asking, are you doing most of your filtering manually in the lab or with some kind of vacuum system in the lab?
Vacuum systems. So Chris has a benchtop pump. So the filtration is done on a benchtop setup. And at NRI, we have a backpack that is a pump. It's a mobile pump made by Smithroot. And we can bring that around and set it up anywhere. And so we typically, for this project, we used it like a benchtop setup. So we haven't done any manual filtering.
Thank you. Our next question is from Chris, and they're asking, as research in this area becomes more mature, is this a process that will need to be conducted by a vendor, or can it be done by citizen monitors?
The sample collection, I really hope, can become a network of citizen monitors. The training needed to collect a sample is really not that difficult. Nobody needs any prior experience. And the downstream stuff is what would probably require more collaboration with groups that have labs that know what to do. But the collection, absolutely, I think, can be citizen science or lake associations or smaller groups.
That's great. Our next question is from Naomi, and she asks, what has been the biggest challenge in the first season of data collection?
Sorry.
I think I would say
Chris.
ISOF was earlier than expected. So we kind of had to prepare and go out. We were trying to go out as early as we could in the spring before lake stratified. So shifting to be able to like go out early and get ready for that was probably, I would say, like something that was unexpected and our biggest challenge. Also COVID was, you know.
Yeah, COVID was was one of the challenges, but yeah, I think like Chris had mentioned, the early season sampling was probably the hardest part, getting out when it was really cold.
Yeah, I bet. We have another question from Nicole, and they're asking, when will this eDNA method be ready for AIS coordinators to use for early AIS detection?
We hope to have the guide, which kind of condenses our recommendations down by soon after our project end. So at the end of next year, we'll probably have preliminary recommendations, and we'll be working with MACER closely to format that into a final guide and figure out where that's going to live and get distributed from. So 12 months, probably we'll have preliminary stuff that we'll talk about the next showcase. And maybe another 6 months after that, we'll have the guide out and a lot more information that people can get and use.
How exciting. I know many people will be waiting for that to come out. Our next question is from David, and they're asking, after a sample is grabbed, how long is it good for before it would go bad or no longer be useful? And a second part of the question is, where would a sample need to be shipped for analysis purposes?
Sure. For our project, we had a filtration timeline of 48 hours. And if something goes wrong or you something happens where you can't get to it, our absolute like drop dead time was 72 hours. And from other literature, it's basically, it looks like the time frame that a raw water sample, DNA remains good, is a lot longer than 72 hours if it's kept ice cold in a cooler that gets no sunlight. And basically that was what we did. Most samples are filtered in 48 hours. Some were filtered within 72. Shipping samples, there's many labs around the state that do this kind of work, and each lab has kind of different experience or different protocols or what they're used to doing for eDNA samples or genetic samples. So yeah, there's capabilities kind of throughout the whole state, I think. Most universities, and there's some private companies too.
Great, that's good to know. We have another question from Natalie, and they're asking, are many Minnesota lakes already being regularly tested for eDNA as part of their zebra mussel early detection program? And are you looking at any of that data?
I'm not real familiar with that program. Most of the eDNA activities that I know about going on are mostly focused in the Mississippi River and looking for the movement of Asian carp species. So I don't know much about the early detection program for zebra mussel that's going on right now, but I'll look into that.